Welcome to this video on translation, and we are going to discuss the location and also the steps of translation. First of all, we want to know that translation is where the cell reads mRNA to produce a polypeptide or protein, and we need a ribosome, tRNA, which stands for transfer RNA, and amino acids for the process of translation. So, among other different factors as well. But anyways, we see here in the cytoplasm of the cell, translation is going to occur at the ribosome, and the mRNA is going to interact with the ribosome in order to create a polypeptide, an amino acid chain. And also we have tRNA right here bringing the amino acids to the ribosome to build that amino acid chain, the polypeptide. So firstly, we want to look at the separate components or the key players in translation, look at their structure and how they work. So we have, firstly, the ribosome structure. So the ribosome, first of all, has a larger subunit and a smaller subunit. The large subunit is where the tRNAs are going to actually bind to, and the smaller subunit is where the mRNA is going to bind to, first at the mRNA binding site. So there's 3 sites for the tRNA in the large subunit, the 60S subunit. The E site stands for the exit site. The P site stands for the peptidyl tRNA binding site. And the A site stands for the amino acyl tRNA binding site. Okay, next we want to look at the structure of the tRNA, the transfer RNA. Here's a closer 2D diagram of the tRNA. So first of all, we can look at the shape. It looks like a T, right, across. And also, we want to look at 2 important locations in the tRNA, right? We have the amino acid attachment site and the anticodon here below. And the rest of the tRNA, right, is going to be basically nucleotides, right? And the reason or why this shape is possible is because of the hydrogen bonds that are formed between the nucleotides or the bases of the nucleotides, right, allowing for this T shape for the tRNA, right? Okay, so that's a closer 2D structure. And on the right is a simpler kind of diagram of the tRNA that we're going to use. So again, we have the tRNA right here, the anticodon, and the amino acid on the top that's attached to the amino acid attachment site. Okay. All right, so first of all, how does tRNA bind to an amino acid, right? So in order for us to put a amino acid, or the cell to put an amino acid on the tRNA, right, it needs to go through a reaction with the help of an enzyme called amino acyl tRNA synthetase, right? So we have our tRNA right here and our amino acid, and we want to combine them. So amino acyl tRNA synthetase is going to interact with the tRNA and the amino acid, go through a reaction that requires energy, ATP, in order to produce our tRNA binded to the amino acid, right? Okay. So here's just kind of a overall diagram of the process of translation. We can see how the tRNAs are interacting with the ribosome and also how the mRNA is interacting with the ribosome. And we're going to go through the 3 steps of translation, right? Okay, so here are the 3 steps of translation. Actually, they have the same name as the 3 steps of transcription, but we want to remember that translation and transcription are different. So the first step of translation is called initiation, and this is when the small subunit of the ribosome is going to bind with the mRNA and the initiator tRNA, which is going to hold methionine. Also, the initiation factors are going to help or facilitate the binding of large subunit to the small subunit, right? Number 2 is elongation. So this is going to start with the codon recognition by the respective tRNA in the A site. And also there's going to be a peptide bond formation between the amino acids, between the tRNAs, and also the translocation of the ribosome, right, to continually read the mRNA. And step number 3 of translation is termination, right? And exactly just how it sounds like it, translation is going to end in termination. And this happens when the stop codon is reached, the stop codon of the mRNA. And this is because the stop codon is going to code for the release factor in the A site, which causes the dissociation of the ribosome, the release of the polypeptide, the release of the mRNA, right? So everything dissociates and translation ends. Okay, so let's look at each step closer. So step number 1 of translation, we have initiation, right? So right here, we have the small subunit of the ribosome, right? And we have our mRNA strand, okay? And we have our initiator tRNA right here. And the initiator tRNA is always going to have the starting amino acid methionine. That's going to, and this initiator tRNA is going to have an anticodon that's going to complementarily bind to the start codon in mRNA AUG, right? Okay, so the small subunit of the ribosome scans the mRNA until it comes to the start codon, and the initiator tRNA binds to the start codon. And that actually is going to cause the large subunit to come and combine with the small subunit, right? And the initiator tRNA is going to be in the P site of the large subunit. Okay. Second step, elongation. Okay. So what is being elongated? So the quick answer is it's the amino acid chain or the polypeptide chain, right? So this is the first step of elongation, right? Okay, so we have codon recognition by the tRNA at the A site, right? So remember, this is the A site right here. So the tRNA right here is going to come over. And so tyrosine, or this tRNA, is going to have a specific anticodon. And I forgot to write that down right here. Sorry about that. AUA. AUA. Okay, so you can see that this tRNA has a specific anticodon, and it comes over to the A site, and that anticodon is going to be complementary, right, to this codon right here, UAU. Okay, and that's why this tRNA was attracted to that site. Okay, number 2 of elongation requires GTP energy, right? and that's for the formation of the peptide bond, right? So here's our growing chain of amino acids, the polypeptide chain, and it's going to form a peptide bond with our tyrosine amino acid that's attached to the tRNA and the A site, right? So basically this whole chain is going to transfer over to the tyrosine at the A site. So the peptide bond forms, the growing polypeptide from the P site bonds to the A site amino acid, right? Step 3, we can see how that all transferred over right here. And now we have our chain of amino acids here in the A site, and in the P site, that tRNA doesn't have anything, right? No amino acids. Okay, so what's the next thing that happens? Translocation. Translocation. So translocation is when the ribosome is going to shift, and this is also going to cause a shift in the mRNA. And this process requires energy as well in the form of GTP. So we'll take a look at that here. Right. So the ribosome shifted right in this case, and that caused the mRNA to shift one left. And what happens is that the tRNA in the A site transferred over to the P site, and the tRNA in the P site transferred over to the E site, which is the exit site. And that tRNA got kicked out. And now we have our growing polypeptide chain, amino acid chain, and the P site, right? So this is going to keep on, this cycle is going to keep on repeating until we reach termination, which is the third step. Okay. Termination, right? So the ribosome is going to keep on reading, right, the mRNA from the 5' to 3' kind of direction in terms of the mRNA. And it's going to stop when we reach the stop codon in the mRNA, which is UAA. And there's, by the way, more than one stop codon. This is just one of them. And it stops the translation because the stop codon is going to code for something called the release factor right here. Okay. Oops. Let's see if this is going to work. Okay. So this is called the release factor. Oops. Right? Okay. So when the release factor binds to the A site, it's going to actually cause a hydrolysis reaction, which means it's going to break that peptide bond that's holding, binding the tRNA to the polypeptide, and it's going to cause the release of that polypeptide chain, right? And also that tRNA is going to be kicked out from the P site, right? And next, what happens is, or at the same time, right, this requires energy, right? The whole ribosome, mRNA, everything is going to dissociate, right, because of that release factor, causing that dissociation. Okay, so that was a kind of quick, rough overview of translation. Again, we looked at the 3 steps of translation, which was initiation, elongation, and termination. I hope you enjoyed this video. Please like and subscribe and follow for more content. Thank you.